Bonjour à tous.
Good morning, everyone.
Et de vous
It
avoir
is
tous.
a peculiar moment, a really important moment for us to have you all here with us. I would like to thank you very much for being here, of making the journey to come here. For all of you who are here in Rome, I am really happy also to know that we are really numerous on the Zoom, so remotely as well. It is an important day because the mission of our association is to create a positive dynamic around the people that have this disorders, and this dynamic can only be created when we are all together. And when we see the professionals and families around here, we have physicians, people from healthcare, students, and professional of the culture that follow and help your kids today. So thank you very much,
Le
Sonia
barbier.
Lecor, Bérénice Pallier, et Marion Barbier-Couchard, and all the students here in Flaubert that give a lot of energy to help the children day in, day out. And through our international connections, I know that we are together in the USA, we are together in Australia, we are together in Europe, we are together in North Africa. We are collectively together today, and I have a really strong thanks to give to Professor Fabre, the coordinator that made all of this possible. It was essential for us to be all together. There are no countries, there are no distances, and it is a pleasure to be all together today. So the challenges for the association is, and its mission is to look at all profiles that are similar and at the same time heterogeneous, and to give answers and follow up for everybody, with a follow up that is adapted to any case, so to be applicable in every aspect of life, because we talk a lot about healthcare, but there is not also only healthcare. Sorry, so it is important to follow up the whole life of the person that has these disorders linked to the MYT1L gene. There is a really strong dynamic right now that exists for now 6 years when we think about the international Facebook group, the Facebook page, which is called Emigrate PL family page. This has existed for 6 years and has seen a lot of interaction. We are at more than 200 people that subscribed on that page, and you know that these are things that families do, so we are more numerous than the actual number. More than 100 families in Europe and more than 35 families in France. The will to develop the association Les Extra-Vaillants was because, first of all, we wanted to have to have a vision to be really together. Today is like we would have a lot of mailboxes and addresses throughout the world, and what we wanted to do through that, to have a common address to put together all the information, because we know that there is a strong demand for families for information. And with the study of Dr. Coursimault, Guénon, et Lecoquierre, we were linked to diagnose of obesity, autism, and so on and so forth. We know that doesn't correspond to everything that the families are going through. Thanks to this study, we finally could fine-tune the profile, and the physicians and experts will continue to study. This allows us to have a common backbone and to go forward in discovery and development all together, and to have a recognized pathology. So the website was one of the main undertakings of this year. There has been the creation of the internet site. We have had more than 530 visitors, more than 1,100 sessions, so people come back, 26 countries connected, to say that we really need to be all together, and we could develop several communication tools, either short videos or we will improve that because we have now the support of a communication agency. And we also opened a French-speaking panel. It was not to separate things. It's only because for administrative reasons we needed, because we have some peculiarities in the French system that would advance us all together if we could get all together on a French level. We are also recognized from an international standpoint, so this is really important, and we are happy for that. We also had an optimization of the logo, because the first logo was homemade, let's say. And we have now a more elaborated logo through a communication agency. The choice of the elephant is because of the community, is all the positive values of community being together, going forward together, and that's why we chose this nice animal. We collaborate, and we collaborated this year with the different stakeholders for rare disorders and disease. I'd like to thank very much the Centre de Référence des Anomalies in Rouen. Thank you very much, Dr. Guéraud and Coursimont, and Dr. Lecoquierre, and also Gothenburg for his welcome. And know that we are really lucky. We are extraordinarily lucky to have a team that listens to us, that follow up on us, that has a will to build something together, and that is quite spectacular. Thank you very much to all of you. With all of them, we started a common mailbox, let's say, approach. We could obtain a code, and this code is really important when then you have some administrative things to do. That gives a backbone and a reconnaissance and a acknowledgement of the disease. We opened the study on food deficiencies, and we were also at the rare disorders day. We are also attached to the I have a binom, Gwendoline Gio. Thank you very much for the organization of this day, but Juliette Coursimault. It is always worked upon collectively with the objective to improve the life of patients, with the association, with the professionals. And I will show you later on through the day a project on which we worked, which is easy to read and to understand. So to present the pathology and having children understand it. So given access to children to knowledge on what they have and what strength they have. And it is a system of continuous training throughout the day and participating into projects. The collaboration is also associated with l'Alliance des Maladies Rares, the Rare Disease Alliance. So we exchange in between association, we help each other, and we continue to work collectively through, albeit keeping our peculiarities. And we also had the support of the Rare Disorders Foundation, allowing us to participate in different projects and to build up for the future for people with this syndrome. The actual dynamic, the current dynamic exists. We see that in the number of publications that continue to increase. We have research in the States, in the UK. We see that these do not cease to increase. So there is really there are really questions asked and a dynamic that we want to continue and to broaden. And this leads to this first incredible day all together with all the stakeholders here to talk about these problems. So building tomorrow is to continue to be relevant, a relevant stakeholder, so that all our family can continue to follow up on the collaboration that have been created, create bridge with on an international standpoint, as well as on the administrative level, research level, and so on and so forth, and also develop project with people in education, with the sports world, because we want to be active and having our patients, our vaillants, acknowledged everywhere where they can be, and give them the means to be all they can be. This is really the starting point of an adventure we will live all together, and I'm really happy to see all of you here and connected remotely, and I thank you very, very much. We're really happy to welcome you today for this day together around the MYT1L gene. We hope that you had a good trip, that you are well here in Rouen, and this was my thesis material, and we were far from imagining that 2 years later we will be all together here with family from all around the world and professionals interested by these pathologies. So thank you very much for being here today. We hope that this day together will give you the results you are expecting. We have quite a complete presentation on the gene and genetics. We will give you some short reminders so that we can level the knowledge of everybody here and connected remotely. It is, of course, quite a busy presentation, so do not hesitate to take notes and to formulate any questions you might have now or later on.
Bonjour à tous. Moi aussi, je suis
Good morning, ladies and gentlemen. Thank you very much for the invitation. What we will talk about today, we will swap a little bit with Juliette. I will talk a little bit more about the lab and the biology and clinical aspects. Here is the plan of the presentation.
Et leur impact. Ensuite, on va présenter
some general information on the human genome and variation and their impacts. Then we will talk about the MYT1L gene and how it works and what it does. Then Juliette will present everything that is known about the phenotype associated kindred to the MYT1L syndrome, then the fundamental research, and then to finish, some perspective for the future. Let's start with the human genome.
Milliards de cellules. Voilà,
in the core of each cell in the human body, we find the genome. This is unique to each individual, and it is like a recipe for the fabrication, for the creation of the human body. It is found in the chromosomes that are represented here, and the DNA is compacted in these helix in the chromosome and created by 4-letter A, C, T, G, with the A going with the G, and so on and so forth. And this creates the nucleotide that constitutes the genome. Our genome is composed of 20,000 genes, so 20,000 recipes. The genes, if we look at their structure, is composed of introns and exons. Exons are the critical regions of the gene. So here we found the code used to create a protein. And so the exome, which is the bulk of the exons, represents 1% of the genomes. The exons are the main responsible, quote unquote, for disorders and these kind of syndromes. And that's why we look into these to try and understand how it works. So these genes give the information to create the proteins of the organism. So it's a sort of a recipe that will be read by the machinery in the body and will create the proteins. One of the basic notions that you have to understand is the variation.
Et donc, on a ce petit pourcent ou ce petit pour mille de...
And we have this percent or per thousand of variation. We do not use mutation that was used in the past. We want to avoid the connotation of this word. So we talk now about variation, and they can be frequent, rare, small, big. Big variation could be, for instance, say an entire chromosome, one too much on one less, and the small ones can be a simple modification of one of the letter of the helix.
Mode de transmission de ces variations.
Speaking about the transmission of such variations, most variations are inherited by parents. So half of the variations that we have are inherited by our parents, and some other variations are called de novo, so they have not been inherited by parents. So these are errors in the replication of the genome that occur and that can occur in different moments. This is a really important notion in neurodevelopment diseases, because this is the cause of very many neurodevelopmental diseases, especially when we talk about MYT1L. So variations can have different impact. This is a summary. Most variations that can occur have no impact. So de novo variations between 50 and 100, most of them has no impact. And when they occur in a specific area of our genome, they are detrimental, so they lead to genetic diseases. In some cases, the impact is even positive in some species. So over a million years, there could be positive variations for some species, but in genetics and especially in medicines, we are most interested in variations having a detrimental, so a negative impact on health. So this is what I can tell you about the impact of such variations. Now, MYT1L, we have identified the cells of the brain system, of the nervous central system, the neuron, and they play a crucial role in this syndrome. Neurons are cells transmitting nervous impulse, enabling communication with the nervous system, so speaking, movement, memory, all functions related to the brain. In addition to the neurons, there are also other cells that we can find in the central nervous system, glial cells, for example, and oligodendrocytes that have a supporting function in the fabrication of myelin. That is this wrap, let's say, that you can find on neurons, enabling and smoothing communication. And then there are astrocytes and microglia. These are two cells with a supporting function for the central nervous system. So these are the cells. A neurogeneticist is the neurogenesis, is the formation of functional neurons that are integrated in the nervous system. And everything happens in the fetus, so in the womb. And so they work as precursors to neurons. They develop into neurons, and then they will be diversified. This is neurogenesis, and this process plays a crucial role when it comes to MYT1L gene. Where can we find it? We can find it in the brain, and specifically in neurons. Keep this in mind. It expresses itself in the neurons, in all neurons of our nervous central system, and we can find it especially in the womb, in the fetal period. So it appears in the fetus, and it plays a crucial role in the maturation of neurons. We know that the gene expresses itself also after birth, especially in our brain, in our nervous central system. But the most important period is the fetal period. What is its function? It plays a role in the fabrication of the MYT1L protein that plays a role in the maturation of neuronal precursors. So it plays a role in the transcription. What is the origin of the name MYT1L? Myelin transcription factor 1 like. This is the meaning of the name like, as you can hear, because MYT1 gene had been discovered before, and so scholars said that it looked like MYT1, but there is a difference with the MYT1 because it just looks like a MYT1, but the function is not the same because it does not fabricate myelin. So MYT1L, as I said, is expressed in the neurons and plays a role with myelin. This protein, MYT1L, you can see it here. So it's a molecule that looks like this drawing, this figure. And you can see here in this representation, the little unities that make up this molecule and their functions. There are different unities that are called zinc fingers, 6 domains mainly. The structure of the protein. So let's talk about that. There are these 6 domains, zinc fingers, that enable to connect the attachment, the connection between the protein and the DNA. So this is the transcription role of MYT1L. This protein binds, clings to the genome to trigger or repress the expression of other genes. It's a bit complex, but this is how it works. MYT1L can change, so it can increase or decrease the expression of some target genes. That is its role of a transcription. It's a transcription factor. We have already spoken about its importance in the fetal period, and the gene can express itself in a specific time and in a specific place of the fetal period. The expression needs to occur in a specific moment for the development of the central nervous system. So it's a kind of a conductor of an orchestra. It plays the role of a conductor in an orchestra because it plants the expression of many genes, making sure that a neuronal precursor becomes a mature neuron. So, as I said, this protein manages all these processes as a conductor at the orchestra. And now I would like to give the floor to Juliette. Okay, as we said, MYT1L was discovered in the 1990s. The implications of this gene has been discovered, are more recent, around 2010. There was a scientific paper reporting and speaking about the diseases connecting to dysfunctions of this gene. And what is the impact? Intellectual deficiency, epilepsy and eating disorders. You can see here the impact on chromosome 2. So what is the situation in a normal chromosome 2 and with the deletion? There is one missing part. So how can we understand where are these, if there are disorders? We use the CGH array. The aim of this analysis is to make a comparison between a control DNA and the patient DNA to understand where are duplicated or deleted regions. Now, ever since 2011, many scientific papers have been released in literature describing and reporting more and more patients with lesions on chromosome 2. Here in this graph at the top, you can see chromosome 2 and in a blue MYT1L gene, while each gray line represents a patient. So thanks to the different descriptions of these patients, we could understand that the gene was responsible of the clinical symptoms connected to chromosome 2. So the responsible was the MYT1L gene. We compared all these patients and what emerged was that this gene was the responsible factor for the clinical phenotype of such patients. After this discovery, more and more scientific papers were released speaking and reporting punctual variations that in the past we would call mutations. And now we're going to speak also about the variations of the sequencing. Right, sequencing variations. We took a look at the structure of the gene and the genome before, and we have already touched upon this topic a bit. So we're speaking about a modification in comparison to a reference genome. So there are two types. You have substitution, nucleotidic substitution. So on the left hand, so a letter has replaced another letter. On the right hand, you can see duplication or insertion. So there is one more in the integration of one letter more, G, for example. So you can see one more G than usual. And you can also see big deletions and big substitutions that are associated with MYT1L, and these variations do not have the same impact. So duplication is not the same as a deletion. Okay, variations, sequencing variations that can occur. There are two important roles leading to a disease. On the one hand, you have missense variation. Here you can see this red dot. So it's a really localized area in the protein, and an amino acid is going to replace another amino acid in a specific area of the protein. And so as a result, the protein is still there, but it had some defects in a specific localized area. And this has a consequence for the functioning of the protein. And we call this variation missense. So that is different from truncating variation that completely disrupts the gene, and as a result, the gene can't express the protein. And these truncating variations are divided into three types: nonsense frame shift and splicing variation. So it can occur. To summarize, missense takes place in a specific localization, a specific area. The second type, truncating, is going to disrupt completely the protein. So with missense variation, the disease is going to be milder. And Dr. Coursimault studied these variations. Now, to summarize the molecular mechanism responsible for the MYT1L syndrome disorder, here you can see the representation of MYT1L. For each area of our genome, you have one copy coming from the father, one copy coming from the mother. So you have two copies of such genome, of such protein in our genome. And when there is a punctual mutation, one copy has a specific mutation, while in the second example is a partial deletion of the gene. So this is a bigger anomaly detected by the CGH array. This the technique that was mentioned by Juliette before. And the third example is a complete deletion of the gene, where you have just one copy of the MYT1L gene that you can find in the patient. And having just one copy that is working, one functional copy of the MYT1L gene is called haploinsufficiency. So there is just one working, functioning copy, and this leads into symptoms. Now, duplications, big size duplications. Let's talk about this, which is a bit neglected. Here you can see a complete duplication of the gene, and the second example is just one copy of the gene, the in gray. So instead of having three copies of the gene, you can have different. So if you just have if you have three copies of the gene because there was a duplication, we do not know exactly what is going to be the mechanism. So for the moment, this duplication, so having three copies of the gene hasn't been studied in death, because so far we have worked on haploinsufficiency that we mentioned before. There is an ongoing study on duplication whose aim is to characterize, to understand what duplications are responsible for each phenotype, and these studies are underway. So we don't have the results yet. Now, what are the techniques that we use to understand, to analyze these variations? We use sequencing. With the patients we work in the lab, we do NGS sequencing, next generation sequencing, and it's the same thing. It has different names, but it's the same thing, next generation sequencing. So we sequence different genes that play a role. So we can work on the panel, on the exome, or on the genome. The aim is to detect the sequencing of the gene to understand what are the variations that occurred. So we could identify thousands of variations, and we try to interpret them and understand how they, what is their role in the disease. And once we have identified a family of variations, we also analyze the rest of the family. So, for example, parents of a child with a variation are analyzed. So we use Sanger sequencing, and which is a much easier technique. So this is the technique that we use.
Pour bien expliquer la notion de panel, d'exome et de génome.
One slide to explain the panel of exomes and genome. Here in our genomes, we have 4 genomes that are represented in the panel. We will decide to sequence only 2 of these because these are the ones that are important in the disorder or disease that we want to detect. The exome represents all the exomes, so 10% of the genome, and then there is the complete genome. And 2 approaches that can be implemented for the NGS sequencing is the solo sequences or the trio sequencing. This is a little bit technical, but the solo approach is to do it on the children and then potentially do a targeted sequencing on the parent or parents. The trio is to do at the same time the 2 parents and the children to see if variations are transferred and to be able to identify possible transfer of sequencing. This is a little bit heavier on the testing and lab side, but both approaches are usable. So the data developed through these processes are really important. There are 5 million variations that are identified on a human genome, 20,000 variation on the exome, and several tenths of hundreds in a panel of genes. So we have a bio, IT, and medical challenge to try to take out the noise from this data and only have the pertinent data for the health of the patient. This is a representation that we can have. We have a series of variants. So you see there is variants on the patient, and all of the other informations are intertwined and linked to that. It is, of course, complicated, but I wouldn't want you to think that it's too complicated. Of course, there are many things that are taken into account in the interpretation. The gene that is impacted, the phenotype, is it frequent or rare, has it already been identified on another patient, is an ex novo or transferred by the parents. All of this pushed the geneticians to try and harmonize our practices in the interpretation of these variations. And start in 2015, some rules have been developed internationally, allowing people to work with the same conditions and parameters. Of course, right now, interpretation is still a challenge, but everybody is working with the same fundamentals and same basis. So that allowed us to learn a lot in latter years on these topics. The aim of the interpretation of variation is to classify variation in 5 classes, from benign to pathogen, 1 to 5, and to create a diagnosis. What interests us most is types 4 and 5 in the phenotype of the patient. Some other variation will be excluded because they are considered benign, and in the middle, class 3 is a variation that has an uncertain significance. We do not know yet if these have something to do with the disorders. We find more or less 10% of the patients in this class. This is a challenge, of course, but most of times we can conclude with tests in exome or genome. An example in terms of interpretation, we will have 2. We will give you 2. This is just to give you a broad idea on how it works in the lab. Here, the patient has 5 years of age. He has a neurodevelopmental disorder and malformation of his hands. And we detect a variation on the MYT1L, and we realize that this is found in 3.88% of the general population. If the variation is frequent, this is an indicator that the pathology is not as serious because it's quite widespread. It has no impact on the MYT1L gene. There again, another argument in favor to the benign effect. It is considered as benign in a collaborative database like ClinVar, and is inherited by the father because we carried out the trio test, and the phenotype doesn't lead you to think to any syndrome associated with MYT1L. So it is still benign. It is not the origin of the diseases of this children or malformation. Of course, it's just a broad idea, but just to explain. And another variation that interests us more, so here a 5 years old children that has a neurodevelopmental disorder and hyperphagy. We have all his parameters, all his information, and we have a clear idea of the pathology of the patient. So if we analyze the exome of this patient, and we find this GLU 813 star, we see that this is absent in the general population. This leads to things that is thus rare. The pathogen also leads to think that it has a strong effect, on the MYT1L gene, and the genetician here is completely sure that with all these criteria that he found the variation leading to the disorder or disability. Without transition, a couple of words on the diagnostic odyssey. This slide comes from the AnDDI-Rares site. It is just to say that there are some circuits that can be complicated to have access to a diagnosis for the patient. There is a time which is most of times quite important, and there are lab techniques that were only implemented in latter years, so not all the labs are aware or equipped to carry out this analysis. We advance on that a lot, nevertheless, and we have the luck in France that we have politicians that support a lot of rare diseases. There is a national plan for AnDDI-Rares to create a center of competence. This is something that is really important in terms of structure existing in France, and also for the lab part, there is the national plan for the rare diseases. We will get to that in the next slide. This is a structuration that was announced by Manuel Valls in 2015, and the objective is to incept genomic medicine in the general system for the identification of genomic diseases. So we have now two platforms, one in the north called Sequoia and one in the south called AuraGen. We collaborate here in Rouen with the Sequoia platform, and this gives you access to gene sequencing techniques that are complete and top notch. And with this restriction of the techniques, we hope in the future to be able to give a faster and easier diagnostic to the patient, diagnose to the patients, and to limit more and more a diagnostic error. That is all from my side. Thank you very much.
On va poursuivre maintenant donc avec le
We will continue with the phenotype associated
On a repris ce
to the MYT1L. We had the impression that there was a similar phenotype in different individuals. There were a lot of data, a lot of papers and articles, but these presented a lot of uncertain points. So we got an interest in this pathology because 3 individuals received at that time a diagnosis, and they had a variation in the MYT1L gene, but they had different phenotypes. We have studied the literature, and we thought it was interesting to assess the clinical data to create new phenotypes and eventually research for a particular phenotype. So that is to better identify the genotype phenotype. We collaborated international with physicians in order to do that through international network for rare disease. We had a collaboration in France with the AnDDI-Rares platform, and then we worked with the US lab GeneDx. That is a really important lab carrying out studies for rare diseases. We contacted a lot of professional geneticians, pediatricians, physicians, and so on and so forth in Europe and in the States. After validation with the parents and families, they transfer data and complementary data with a complete anamnesis of the patient. So we wanted to make sure that the variation that we had identified was pathogenic and responsible for the phenotype. So we needed a lot of data in order to carry out an assessment and an interpretation that was precise. And we have included 40 patients and excluded 30 patients for which we didn't have enough information. Here is a representation of the patients that were included in this study: 9 in the States, 28 in France, then Belgium and Luxembourg. The average age was 11.3. And there was an analysis that was done in exome or genome or a panel, and we didn't modify the inclusion characteristic in this study. Then we looked at different elements: the obstetrician period, the neonatal period, the psychomotor and neurologic development, the behavioral disorders, and so on. And if we look at the elements that we have identified before the birth, 30% of mothers had problems during the gestational period with a fetal malformation for 8%. Of course, these were not specific to the MYT1L gene, but they were more or less severe, so we couldn't identify this pathology at that point. In the neonatal period, 35% of the cases, they would be born at the end of the time, or have a delay in development in motor or language in 100% of cases. With a seated position at in average at 11.5 months, and then walking in between 22 and 28 months. There will be a difficulty in fine motricity by 81%, and hypophony for 47%. Most of 95% of the children will have some problems with language development. Not all physicians could send us really precise information about this. We, of course, couldn't push the study really much forwards on this. Some had major severe language problems. Others were able to construct phrases and sentences. Then these children have evolved with some intellectual disorder, 70% of them. 14% we didn't have enough information of the severity of this deficiency. And what's interesting in this study is that we showed that 30% of the patients didn't have intellectual deficiency, but they had learning disorders with dyslexia, dyscalculia, and so on and so forth, dysphasia, and they needed a re-education in 84% and 94% of the cases, and they needed additional help in small classes or in a normal class, but with a particular aid.
Okay, there are medical and paramedical cares, care treatments that can occur. So different professionals, neuropediatrician, endocrinologist, genetician, ophthalmologist, different kinds of professionals that intervene in caring. and giving support to these children. Almost all children have behavioral disorders, unique or multiple. The severity is variable, but very often invalidating, so having an impact on the daily life, very often 50% of the time with autism, intolerance to frustration, aggressivity, and ADHD. And 23% of people also had epilepsy, all different kinds of epilepsy. So focal crisis, EEG anomalies, and epilepsy had different level of severity, and it started in a neonatal period, so during childhood. As for neurological disorders, we noted that patients could have disorders related to coordination, unstable balance in 7 people out of 40. And then we could also have the MRI scan of 29 out of 40 patients. And in this MRI, we realized that there were anomalies in 28% of cases, not very specific and not severe, and other controls had been carried out after. And there was nothing specific, there is nothing specific related to MYT1L. And while some elements could have been related to this pathology, that was not the case. As for eating disorders, in this study, it emerged that 1 out of 2 patients were normal, and 58% of patients were overweight or obese, and the body mass in adulthood was 28.6. This is the median, and the weight phenotype was associated in 45% of cases related to overeating, so hyperphagia, eating too much, eating too quickly, or snacking all the time. And what also emerged, what was reported by very many scholars, was that 18% of patients, of children, had it was hard for them to gain weight, vomiting, and so these patients were not too thin, but it was simply a bit different for them to eat, to be fed, and they had hypercaloric diets. We put together data to establish a growth curve, and what emerged from this analysis is that patients were gaining weight in a premature age, about 3.5 years old. And we realized that when parents started really checking on the habits of their children, so snacking, they were checking, they were trying to support their children, well, the situation would change. So children would go from being obese to being overweight. And then there are also associated ophthalmologist disorders, astigmatism, strabismus, also sleeping disorder, 33 of cases, difficulty in falling asleep, and also OSO, so sleep apnea, that is associated in overweight children. Endocrine disorders, well, that is not very frequent, so 10%, it is not specific. And then short stature, so this was observed in 8% of cases. And we also analyzed the morphology of the face that can also be related to some genetic diseases. So different aspects of the face are studied to find a sort of correlation and a resemblance among children. There were some characteristics, for example, slender eyes, anteverted nostrils, but these characteristics were not constant and were not specific. So you can't really correlate these features with the disease. So thanks to this study, we could confirm elements that had already emerged in previous studies, for example, intellectual disability, premature or overweight, epilepsy, and also secondly, ophthalmological disorders, sleeping disorders, and 43% of children had a normal weight. And then a new elements, 30% of patients had no intellectual disability, but they had learning disabilities, which required a different treatment. And then we could also note that parents checking on their children and restricting their eating habits, that was really effective. Then we identified 3 de novo variants, and 2 variations had been inherited by parents, and they were symptomatic. In 2 patients, we had no possibility to have a sample from their parents. Now, in this graph, you can see the same information as before of the protein. It is flat. In blue, you can see truncating variant, and in red missense variations. So when you take a look at the variations also in this graph, you can find the same results as previous studies. So missense variation are segregated in the 2nd and 3rd domain of the protein. So in the center, while truncating in red can affect any area of the gene. These are the 14 missense variations and 26 truncating variations. So we identified some missense variations in the 2nd and the 3rd domain, zinc fingers domain. This is another version in 3D of the same information. Truncating variations can affect any area of the protein, while missense affect just the purple areas. So we studied a bit more in depth the relationship between a genotype and phenotype. So missense variation, is it responsible of different phenotypes different from truncating? More severe, milder, the same. So we made a comparison of based on 44 criteria, and we didn't find any significant difference. So there is no correlation, there is no difference between missense and truncating variations. And now to finish about phenotype, in 100% of cases, there is intellectual disability, learning disability, obesity in 60%, eyesight problems in 30% of cases, and then behavior disorders in 98%, epilepsy in 23%. And now some information about research. Fundamental research.
Alors, sans transition.
Okay.
Donc là, j'essaie de schématiser un petit peu les...
I will try to summarize what we can do in research on rare diseases, what are the possible steps to be taken, well, the situation changes for each disease is different. The first step is the identification of the disease, characterization, clinical characterization, and then understanding the responsible genetic variation. Step number 2, biological characterization. So what are the biological mechanisms? Why such variation is related to a phenotype? And then in the next step, we work on models, for example, cell models, so animal models, to understand what are the biological consequences. And then step number 3, that is the target. So what are the potential treatment? How can we have an effect? How can we be effective, an effective treatment? This is what guides us in step number 3, what are future therapies. For MYT1L, we are in step number 2. So to understand, we are trying to understand how this disease works.
And the idea to try to go forward to understand the disease mechanism is try to modelize the disease. What I'm trying to explain here is that we cannot have patients to carry out analysis as we would like. For instance, we cannot include several times the same patients, carry out several tests on them. And in order to avoid all that, scientists try to create a model to replicate the disease and try to replicate the disease in some cells, so they will be able to understand how it works on a cellular level. The same goes for the animal model. We use, for instance, mice have MYT1L like all mammals, and we can try thus to replicate the disease in mice to carry out standard tests on several samples that we can thus get and understand what happens in their brain, in their tissue, and in the fetal tissue as well, with repeated measures and so on and so forth. This allows us to have a lot of precise data on MYT1L. Is this completely similar or equal to what happens in humans? Maybe not completely in mice, but anyway, it gives us important opportunities to understand how things happen on a molecular cellular level. This is the theoretical introduction to my explanation, and in practice, in latter years, there have been some major studies on mice. There are 3 international groups that produced, they didn't work together. Each of these groups worked separately, so several groups working on this topic and generated mice with this pathology. And so the groups are the following. There are 2 groups from the US and one, I think, is Chinese, working on this topic. And they could detail with a good level of precision the behavior in mice, anomalies present in their brain in several phases of development, antenatal and postnatal. There are several points that really look in the phenotype, and it's quite promising to characterize and identify this disease in the linked biologic ways. I will not get into the detail of these studies, but a couple of words on the future.
sur le plan comportemental aussi, et essayer un jour peut-être d'aller vers la
And
phase
we will
3,
be able one day to
notamment sur
devise potential treatments, for instance, with behavioral disorders or for disorders if we manage to identify it and treat these phenotypes in mice, that will give us some important leads to develop some protocols for human. I will leave now the floor to Juliette for some perspectives for the future. Just to finish, we wanted to get back a little bit on everything that happened since the creation of this project. There has been the creation of the association Les Extra-Vaillants, the website, the implementation of a explanation pamphlet when families receive a diagnosis of this kind of disease, videos, detailed videos on the syndrome. We also are in the process of creating a reference on good practices for physicians for the identification of this pathology. And we also created a protocol especially for food-related, disorders. This is after the identification in our study of patients overweight or obese. We thought it would be interesting to characterize this better to see if we could link it to other disorders. That is interesting for all patients that have problem of dietary disorders, being overweight, obese. We've done that with several physicians working at the reference centers for Prader-Willi disease and with the Center for Dietary Disorders in Paris. They are helping us to identify, to interpret all results we get on these patients. We started to send questions to geneticians that had followed patients in the primary study, and we are continuing this process. The idea is that families can fill up these forms, these documents, send them back to their physicians and genetists, and they in turn send them to us, allowing us to advance on the molecular genetics associated to the patient. And the objective is to identify some behavioral profiles in order to be able to propose a better healthcare and even therapeutic solutions that are based on other diseases presenting dietary disorders. We have here all the biography and the literature existing on the MYT1L gene, and we have finished. We would like to thank you all for being here today. Also, thanking all the people listening on the Zoom connection, all the people that participated in our studies, families and collaborators. Thank you very much to the Extra-Vaillants Association as well, and to Gwendoline Dio for the organization, and the Rouen CHU for the logistics. We are ready now for your questions if you have any. Thank you very much for your attention.
That should be fine. Thanks a lot for the invitation. I'm so glad to be here with you today. And I am going to introduce myself shortly. Soizic Maingant Le Gall. I'm the Northwest Rare Handicaps Relay Team pilot. And I am also the director of a service that works with people with different kinds of disabilities. Today, I am going to speak about a project that is called Équipe Relais Handicap Rare. So it's about rare disabilities. The aim of this team is to give services throughout France, including DOM and TOM. So regardless of where you are in France, you can get in touch with this team. The mission of this team is to streamline care for patients, so you can ask all questions about what happens after the diagnosis, with whom I should talk, what are the resources available. And that's the same for children patients or of adult patients. So you can get in touch with this team to have support and to have more information, to know more about disabilities and the resources available in your area to meet the needs of your children, or also to meet the needs of families more in general. So do not hesitate to get in touch with them. There is a website where you can find all information. Here, you can also find contact for our service in the northwest of the country. In case you need more information, please let us know. All the teams are related one with each other. We do not perhaps we can't answer to all your questions, but we are here to help you finding the answers to your questions. And we also work in connection with healthcare professionals. So what are the rights of patients? How can patients enjoy their rights? And you can get in touch with the team without any notification. So it means that, for example, even though your children hasn't been really recognized, I mean, the disability or the pathology hasn't been recognized, you can get in touch with the team without any problem. And the service is free of charge for you, but you pay them as taxpayers. We all pay this service because we are funded by the state. Today, I would like to give you some information about tools, about how to make choices in a world that we do not know. Everything is new because of the world of disabilities and health care world is not known to everyone. So the mission of the team is to give information, to help you make decisions, to give you all information to make a well-informed decision. I am going to give you some examples. These are real situations, but this is just to give you an example of situations so that everything is clearer. You will not find all information. I mean, you will not know everything that we can do as a team for you. So there's much more than that. First situation, first example, Juliette, she's 9 years old. She comes from another department. She arrived to our area. Her parents know that she has a genetic anomaly. So there is a follow-up, a consultation, genetic. She has an intellectual disability, behavioral disorders, motor disorders, and she overeats. She spends all day at home. She has no activity, and in southern France, she goes to a hospital that is called the FAR, and since the family has moved to another area, we got in touch with this hospital and also because this center had the contact of this family. The family told us that it was hard for them to make decisions because they were a bit overwhelmed at everything that they had to do. So they didn't know exactly what to do to help Juliette. There were many, many people that they had to get in touch with, and so it was very hard for the family. So after talking to the family, we found an agreement. We told them what they should do, and the family agreed with our approach. And we decided who should do what. So should they do everything on their own? Should we were we supposed to help them? We made an agreement. The first step was to get in touch with people who would help Juliette in southern France to know exactly what were their difficulties, what was their potential. Then we supported the family also in administrative procedures. The first thing was to transfer her file, MDPH file, to the new department in which the family was working. And then next step was to find a place for Juliette, so that they can also have some time without Juliette in the family. So some place where Juliette could spend some time. And then the next step was to tell healthcare professionals to give them information about Juliette. Second example, Emmanuel. He's 19. He has Prader-Willi syndrome, and he lives with his family, with his parents. He stopped going to IMA for 6 years. This center is in difficulty, so they can't help you. So Emmanuel spends all his days in their family. The social worker got in touch with me, the social worker works in a center specialized in nutrition. So the social worker got in touch with us, and we got in touch with the family and with the healthcare professionals.
The needs of the family was a need to rest, and also to find an adapted follow-up for Emmanuel, answering to the needs of Emmanuel, and also to take stock on the rights Emmanuel had and his family too. After talking with the family, we got together, we agreed on the approaches to have, on the different options available, on assessing the needs Emmanuel had and his family had. Today, Emmanuel is 19 years of age. He hasn't the same need he had when he was followed by the institute. To link with the MDPH and also to link with the ESMS that could answer to his needs. And finally, and I want to say this is the longer and part, is to accompany his family and Emmanuel through healthcare, either with options readily available around Emmanuel's households, but also linked to the life expectation, Emmanuel. Third situation is Maxim with Prader-Willi syndrome. He's 24 years of age, and he would like to live alone. He's working in ASAT and is currently living at his parents' house. Our intervention had been suspended, and Maxim family came back to us. There has been some time to understand what their needs were and what were the obstacles in respect to his project. Maxime wants to live alone. There is a worry for the possibility, for Maxime's possibility to manage alone his intake in terms of diet. Parents needed to receive further information to know if they have to do something administratively or not. And also Maxime wanted to have a sports activity, but he had difficulties to find an association or an institution ready to welcome Maxime and his particular needs. So we accompanied them in searching for a place for Maxime to live that would present everything that Maxime would need. Is not because Maxime has a disorder that he doesn't have to normal access to the housing schemes. We supported Maxime to look for sports activity with a installation that would be adapted to his case. We also linked them with the local association for people with Prader-Willi syndrome. We also had contact with the social assistant of the ASET to see if they could if Maxime family could receive support from them. And we also worked on the possibility to assess Maxime's skills in respect to his life project. And we supported Maxime and his family in obtaining the aids necessary for Maxime to receive a house, an apartment. This gives you just an overview of all the things that we do. In terms of access to one's rights, the necessity is to speak the same language, to understand and to agree with everything that we describe on every part of the process. Access to rights, in this case, this goes through a request through what's called in France the MDPH, and there can be some fuzziness, some confusion in what the MDPH expects from a dossier, from a file. The paradox that we have is that in France, to have access to your rights, you have to have medical certificates and also to describe the difficulties on a on normal day's life. So there is a question of pathology, a question of the difficulties you experience in daily life, and that's where the MDPH, which is the departmental institution for handicapped people, that was what is called in France. In respect to this, there are several tools that I found. And maybe you know them already, and if that's the case, I beg your pardon, is just because I want everybody to have a comprehensive knowledge of what's available to you. This is a practical pamphlet, is accessible to everybody in France or elsewhere and this guides you on creating your request, your demand to the MDPH, or also called MDA, is not the same name everywhere. But these pages are really well done, and depending on what you want to ask or what you wish to ask, you can look if it's for a renewal or for a new demand. And you have all the explanation here. And what I wanted also to present today was a tool that is completely free. You can use it as you wish. You can use it to transfer elements to complete the file at the MDPH. This tool that I will present has the merit, and I'm not the one saying that, the family that used it say it. This tool makes you understand quite well what the MDPH demands and asks for. You have a white page which is called Life Project. And of course, you can imagine that this is quite difficult. It is difficult to describe your life project when you don't know how to do it. So we work with families, with Organization for Rare Disease, with several stakeholders. We worked on it several years ago. I think we started in 2016 to create a document allowing anybody and everybody to create such a document easily and understandably. It has a name. You have a first page that explains to you who can fill it up. Anyway, everybody can do it. How is that used? To describe the difficulties on daily life that you have. This is the end of the first page. Of course, I will not describe everybody, but you have a code with a letter A, B, C, D, E, and for each of this letter, you have a description. For instance, A is without any difficulty, B is with some difficulty, C is the person needs the help of somebody else, D, the person doesn't do it, and E, I have no opinion. And then you have several items, for instance, standing up. For instance, here you see everything linked to mobility. For all these items, there is, at the end of the document, and you will see that on the slides, a glossary defining what each item means. Let me give you an example. If we take, for instance, a children of 7 years of age, and one of the items on this page is driving a vehicle. And so you would say to yourself, well, a 7-year-old kid, I can put, it doesn't do it because it doesn't drive. But when you look at the glossary, you see that driving a vehicle concerns any and every kind of vehicle. It can be a scooter, it can be a wheelchair, it can be any kind of vehicle. So this glossary is used for this, to identify what we mean for each item and to give you a precise definition to what is asked. Beyond the columns and the spots to tick, there is on the right-hand side a free text part where you can give a complementary description. Sometimes you have a hard time ticking one slot and you would like to tick both, two of them, or in between of them. And so in those cases, you tick one box and then you give a description. For instance, you can describe the kind of aid this person needs. For instance, to transfer, you would say B because the person does it with some difficulty. But then you would say, hey, because certain things the person does without much difficulty. So you can specify and describe all of that on the right-hand side. And you also have to describe the technical aids the person receives in order to perform some tasks. So I put all the form here. Of course, the link must be on the slides, but we will also give you the link through the chat. Some MDPH will ask to the family to fill up this form when they need complementary information. You have several activities, mobility, communication, simple housekeeping, healthcare, whatever else. And you can add a complementary item if you so wish. For instance, you lack some elements to describe the difficulties your children or adult is experiencing. So you can thus decide to add an additional item, but we would advise you to put a precise description of the item you're adding. You can also ask the healthcare professionals that are supporting your children, or in any case the patient, to tap in and to participate in the filling up of this form. The activities concerning communication here, activities concerning housekeeping and daily life. Of course, this form is for any age. So depending on the age of your of the patient, of your children, some items might not be pertinent. But anyway, we arrive now at the part in the questionnaire where you can describe what, in your opinion, is making the situation of your family or of your children more severe. There are a number of examples, but let me say, for instance, that it is a single-parent family or a family with several children with disabilities or disorders, or the fact of not having an adapted household for a particular mobility problem. All of these things you can also inform here. And I would add as a compliment that one of the greatest difficulties that the families declare is the difficulty when we ask something to MDPH, when we make a request to the MDPH, is to understand one another and try not to see things too negatively when every day you try to be positive and to look at the potential of your children. Despite all of this, since MDPH purpose is to give you the best and the best tailored aid to your needs, you need to be able to describe things as they are and just give the facts. And anyway, this is a difficulty that we have experienced with several families. No family ever told us that it had no meaning to fill up this questionnaire, and many of these families have told us that it was a lot easier to fill this form instead of writing a text on the life project section. And then there is this part that is the timetable. You have the several days, all the days throughout the week, and it is important to describe as finely tuning as possible everything that happens in the daily life and every moment, occasion, or thing on which your children needs support and what kind of support. This allows also to highlight sometimes the fact that you do not have enough time during the day to move to one activity to the next when you need to go take your kids from school and need to see some healthcare professional, and then rehabilitation, reeducation, and so on and so forth. All of this can highlight the difficulties that you are experiencing. For instance, if one of the parents had to stop working to do this full time, you can put it here. This is one last part that is more for healthcare professionals. I don't think it is the best part of the document because in my from my point of view, parents know best what their children need, but this part exists, and it's not because it is written that is for healthcare personnel that you don't have to fill it up if you have something to say. And here you have an example of a filled form, of the glossary of the definition of the different activities, and all the items are described here. And I'd like to finish through this slide, highlighting the actions and things that we can propose on the territories of Normandy and Hauts-de-France, but other actions are proposed by relay groups. The needs are expressed by territory. For the action that we propose, these are open to any family and free, of course, for the families taking part. For healthcare professionals are not really expensive, but for families, these are free. So we have actions in alternative and augmented communication, everything that is communication to strengthen the language or to express oneself when verbal language is not available. Then there is a management of there is an obligation for having field teams and healthcare professional. A second work group is on the management of problems, something that we talked a lot about since this morning. These training are depending on the kind of pathology. These are training on the fundamentals of these problems, on alternative and augmented communication, and the management of problematic behavior. We also have something on the raising awareness on cases of severe epilepsy with 3 associations like Epilepsy France. We created a study that dates back a little bit on the all of our territory on how to support and accompany a person with a severe epilepsy. We can also develop training on multi-dimensional assessment. This is more for professionals, but is not closed. And if we have a group of parents that would like to work on that, or associations of families that would like to take part to one of these courses, we can do that. We can also create awareness raising campaigns for professionals, but for associations of families as well. We have done it with the Prader-Willi syndrome, with Prader-Willi Association Syndrome Association France. This is not, many of these training are not specific to healthcare professionals. Sometimes a group of parents are interested in this. And these training are developed in the Rhône-Alpes region. I have finished my intervention.
Voilà, donc je vais vous parler un petit peu de ce que j'ai pu observer.
I will be speaking about what I could observe throughout my career, what I could understand, what I could bring to your children. This is taken by my own professional experience in kinesiopediatrics, where there's some little patients that I have the chance of supporting, Alice and Silas, and I also made the link of some things that we can find in other pathologies, and well, I tried to devise some protocols and systems. Thank you very much for the psychometricians that sent me some data and allowed me to have a cross-sectional research. I'd like to thank Mrs. Salomone for everything that we could do with her daughter as well. And I also received a lot of information for a lot of your children, many of your children, sorry, and we also try to boost one another forward. I will give you some practical appraisal, and then I will leave some time at the end of my presentation for questions and answers, because that's the most interesting part, I believe, for you and for myself as well. So the psychomotricity troubles that we can find are muscular weakness and hypothonia. We will look at on how we can handle all of this, but this hypothonia is present from the start, and of course we need to fight against it. Of course, we will not be able to solve the problem. The problem will always exist, but we will try to have your children not experience anymore the downsides of this. We will also find some deficit in terms of coordination, instability, difficulty to stay in place, to keep a position, a stable position without moving, and this generates a difficulty of performing precision movement, fine-tuned movements. Let's use an example: the gesture of a tennis player. If we're not stable whilst doing it, it's difficult to do it. If it's already difficult to be seated correctly, writing while seated is even more complex. Let's imagine that you try to write something precisely whilst you are sitting on a ball. So working on fundamentals on stability allows to have an impact also in fine-tune mobility, as you see on the slide. And then you find a disorder in proprioception, that is deep sensitivity, what we can sense, what we can feel. For instance, feeling that my feet are touching the ground, and I am pushing as much on the left foot that on the right foot. The position of my head, for instance, that my head is standing in between my shoulders or tilted slightly to the right or to the left, that kind of thing. And then we will be working also on balance. I wanted to give you some general information to show you, to give you an outlook on how everything is connected in the children and in a baby. For instance, you see that the motor development will nourish the intellectual development, because babies will start to move. He will finally be able to open the drawer, the bottom drawer, and he'll be able finally to get everything outside it, to break everything. Whilst the baby that doesn't have this opportunity cannot start exploring and making new experiences. If your children has severe hypothonia, he cannot play with his two hands because it's too difficult for him to put them together and to put them apart again. But then the discovery of his hands and how they work will not happen as rapidly as it normally would. And let's think about when they start putting their hands in their mouth, they discover things, they discover new things, things are developed on an intellectual level as well. And that is why it is really important that this baby gets in the right position so that hypothonia will not impact his development too much. And children that that doesn't have any intellectual appetite. Let's take an example of a blind child. Sometimes we have some slow development in terms of intellectual abilities because there is a part of the experiencing that is lacking. For instance, he doesn't see the objects near or far from him or her, and he is not curious to go and look for them, to move on his own, to go and look for these objects and touch them and feel them. So we need to find solutions to bypass all of this and to avoid having development disorders. Of course, you have seen that, and you know that children already try to bypass what they cannot do. We talked about motor disorder, but there is also something to say about sense disorders or on sensoriality.
Agréable. Mais quelquefois, certains enfants.
For instance, if I have a teddy bear, a fluffy teddy bear, children can discover something, can feel something they can become keen to, or otherwise they discover they do not like the feeling that the teddy bear gives. If I'm not clear, please do not hesitate to. I'm trying to cram all the messages I'd like to convey in a half an hour. The problem is that sensory disorder will have an impact on the way your children will use their body. And then let's talk about support from a kinesiotherapist or a physiotherapist. You have children who have hypersensitivity, impulsivity, and also a lack of attention. There is a motorbike crossing the road, there's a sound that they hear, and they lose focus and attention. And sometimes for instance, I ask them to raise their hands a little bit higher and they do not want to. So each time there is a lot of patience to be had, and we need to develop these exercises through time. But the kinesiotherapy session, if I am losing patience every time they lack of attention, we will not get anywhere. And sometimes they can have a sensory overcharge or a sensitivity overcharge. So it's up to me to manage all of these aspects because they can be hard negotiators, babies and kids, you might know. And I always tell them, if I'm asking you to do this, it's because I believe that you can. And so slowly but surely we go forward and we go on. That's why that in the principle of the taking care, it is paramount to work individually. It is impossible to work in a kinesiotherapy room where there is some person with Parkinson's doing his exercise, another person running. You need to be alone to be individually focused on the children, one therapist and one children. And I always start with really clear instruction, and I tell them we're going to do this once, twice, 3 times, and I put everything that I can so that we can move slowly but surely forward. Let me give an example. If, for instance, we have to get to the ground and stand up back again, is what you could call a squat, but we would imagine to pick up something and then to throw it away. And I will tell them we will do it once, twice, or 3 times, and at the same time I ask them to count them on their fingers, which means if they can do it once, they can do it 10 times. I will to be talking about strengthening the muscles afterwards, but if we want to strengthen the muscle, we need the children to be able to do an exercise several times. So it's up to us to adapt to the children we are facing. What I find really passionate is the fact that each and every one of them is unique. And it is important to understand that sometimes things are a little bit difficult, sometimes things are frustrating for them, and sometimes some outbursts that they can have is part of their disorder, and they have to learn how to live with them. And it is up to me as a therapist to give them a reassuring environment, a calm environment, and understanding that this can happen. So on babies, I think here you all have older children, but the care, in my opinion, has to be as precocious as possible. As soon as we highlight, as soon as we see some slight delay in the motor development, we have to intervene. That can be how to turn around, how to sit down, and then we get to critical phases. As the more we give the children the ability to be seated in between 6th and 9th month, what we call the normal phase, normal age, even though I don't like this term, but anyway, the faster we manage to have them acquire these things, the more we will be able to advance. Then there will be a lot of compensation, adaptation, of course, but it is important not to wait too much to stimulate the baby. Everything that we do as early as we can will bring fruits, will bear fruits afterwards. When I work with a baby, I am there, he is in my hands, and I will envelop him, and that's how he feels that there is a tonic envelope around him. But I will move faster through these slides to talk about older children. The support in kinesiotherapy is essentially through sport working against hypothonia. We will be talking about scoliosis afterwards because I think there is more scoliosis in these children if compared with the general population, let's say. So strengthening the muscles. We can propose in what I call intensive, which means that if we have 20 minutes, we give our all in 20 minutes. We try to be intense for 20 minutes in the exercise, and studies showed that twice an intense activity twice a week for 20 minutes, which you would convene is not really much, would give advantages and benefits: more endurance, more stamina, less fatigue during the effort, and a longer resistance. So what is intensive for me or for them cannot be the same for you. Sometimes it's difficult or really, really difficult. We can be out of breath. We can have repercussions the day after. This is not a problem. That means that the muscles have worked. If I don't have the impression that I was exposed to an effort, maybe it means that I didn't put too much enough effort in my exercise. And these intense exercise allow to develop muscle fiber, and so those of you who do who are used to sports, you know that this is the way forward. This is the way that we improved. I am used to film, to record some parts of a session, and it is really interesting to see the difference 6 months apart. Something that was really difficult 6 months prior, it becomes more natural as time goes by. We do not have targets or objectives to start with, but the progresses are visible. The idea is to start with simple exercise, easy to do. There I put the steps there in the pictures, because sometimes we can do a whole exercise only through steps for strengthening the muscles. If you have taken steps a lot, you know what I'm talking about. Then I will work everything that is squats and seated and standing up position. You know how difficult that can be, so we will start with I am sitting on a bench and I stand up, then I sit down, and then I stand up, and I do it 20 times, 30 times, and then I lower the bench, and then I add a support with a kilogram more on my shoulders. If I manage to do it for 20 minutes, it means that is not too difficult. Maybe we need to reduce the time, but increase the difficulty with some additional weights, with a lower bench. But everything that we can already do with the weight of our own body is brilliant. There's no need to buy equipment or stuff. There is also a problem on overweight in this pathology, so we will work in a global manner. We will try to work all the muscle groups. For instance, an exercise that we do quite a lot is getting down on the floor on the belly and standing up, and so on and so forth. I can tell you that it's quite difficult to do it if you try to do it 20 or 30 times as fast as you can. And when I work with children, I try to put a little bit of competition to help them. Then, of course, there's exercise, and also with we work with the rope. The rope is really useful to work on coordination, on rhythm. It is quite a physical exercise, and then we can do it with other people, and in terms of inclusion, that's really important. So I work with children, and children play. So we play for 30 minutes whenever they come to see me to a session, or we can have several workshops, and we will move from a workshop to another. I will use protocols, sometimes like protocols that you find in sports, Tabata or EMOM. For instance, in Tabata, I do squats, and I go down 10 seconds, and then I stand up 10 seconds, and so on and so forth. But there you work, you know. These exercises sometimes are too repetitive. They do not like them too much, but I have a colleague that worked a lot with children and developed and devised a lot of games, and these games are really fun to do. You can even do at home with your children. And for instance, you see an example there. It's the principle of a table game of and you throw the dice, and you play with your children, and you have to play with the children. There is no reason that they have to be the only one doing the exercise, so you do them with them. It will not do you harm.
What else do I use? For example, on my phone, I set the time 2 minutes, and I said, we are going to do the exercise for 2 minutes, but this is really abstract. But they can see the flame that goes forward, and it is going to trigger the missile, let's say, and so the fireworks will come up. So it's fun for them. And then you can say in 2 minutes, you will try to do 10 or 20, or next time even more. So it's really important to have a progress, to do more and more. David Guédin is somebody working a lot on brain paralysis, and I have put the link to the YouTube channel of David Guédin. So if you go on his YouTube channel, you can download all the activities that he has with the monsters. It's a sort of monster memory. So each time you turn a card, there's an exercise to do, for example, pumps, different position to make, different exercises to be made. Most children here do not have a specific difficulty on us with a specific group muscle. So the way in which we work is to have a global work, global exercise, working on all muscles. And sometimes it's so quick that we don't even realize that we are working, we're doing a real job. So what are the benefits of muscle boosting? Well, it's a compensation for hypotonia. Their muscle tone is lower than standard. But thanks to this activity, this sort of working out, they can compensate this weaker muscle tone, which can help in their daily life. So they are going to improve resistance to effort, they will get tired later, and they will be more stable, and the hand will be more precise, so for fine motricity, motor skills, and they will improve balance. They will be able to fight against overweight because they're going to burn calories, and it will be beneficial also for their mental health, and they will be happier. Now, when we work on balance, first of all, you should understand what are the causes of balance disorders. One of the causes is hypotonia and muscle weakness. For example, I don't know if you've ever had plaster for 1 month. After 1 month having your leg in a plaster, you will see that your muscle is weaker. You need to start working again. So perhaps balance-related problems are related to hypotonia and also weak muscles. And there could also be problem linked to cerebellum or proprioception-related problems. In order to work on balance, you need to work on automatism. So sometimes we can work on unstable platforms, trampoline, or we can work on the floor, we can work on a carpet, many different activities. Everything is possible. We can work on a skateboard, on a scooter, a motor circuit. And then what I tell parents is that the gym is not, what I tell parents is that is not enough to work in a room, in a kinesiology room, because you need to go to the park. The best thing is to propose as many experiences as possible, different kind of environments. Then balance can be weaker when you're tired. There are specific conditions. You need to bear that in mind. Then working on coordination. Well, I don't do that very much because we don't really have the time, because one session goes really fast. we work on ergotherapists or other professionals, and together we work on visual manual, bimanual coordination, on actions that you do every day. And the aim is that a gesture becomes automatic. For example, grabbing a glass of water. The quicker it is, the easier it will be. And so it is really important to repeat the action many, many times, and it becomes spontaneous, automatic. And some actions are more harder to work on than others. Working on coordination requires attention, and this attention sometimes is lacking a bit. And that is how we need to find ways to have to make sure that the child can focus. So, for example, we work with balls, or with rackets on circuit, or we also do the game of the statue game. We need to be really creative. And then let me also talk about orthopedic surveillance, because I have the feeling that there are much more cases of scoliosis in comparison to the general population. So it means that the spinal cord is altered in the 3 plans of space. Scoliosis need to be certified by your kinesiologist and also by yourself. Well, it means that you as parents are those who stay as much time as possible, your children, and you can see the evolution. So what can you see? What should you pay attention to? For example, on the right hand, you can see the scan of scoliosis. What can you see in your child? For example, one shoulder is slightly higher than the other, or the 2 triangles that create the trunk and your arm is so one is bigger than the other, or the line of the hip is slightly altered, or the spinal cord is not really straight. Or you can also ask your child to bend down, and you can see a sort of bump that appears. So you are the one who should pay attention to these factors, and you need to talk to the doctors in case you see some of these examples. So it is a good idea to have a check once a year, and even more often when during teenage, because children go really, really fast. And then if needed, you could also talk to an orthopedic surgeon. And when this scoliosis reaches 21 degrees, it is really important to use corset, and also to go a bit further in case the situation is worsening. So the main message is that we need to be really careful. We need to have a constant surveillance. So what can we do as kinesiologists? Even though we have if we have a strong shoulders and a strong back, scoliosis can still come, and scoliosis resist. And so even though we do some workout and we work on a muscle and shoulders, we can still have it. But it's a good idea to work on the back and the shoulders. So once again, muscle boosting is really important. And when scoliosis is already there, we need to work in an asymmetric way. So your back is when your back is curved, we need to work on the back to make it a bit slightly straighter. I put my head, my hand on my head, and I say, you need to think that your hair is going up. So I ask children to sit down and to make this movement. This is really important. And it is really important to work on specific exercises and to have a good, to be coupled with breathing. Kinesiotherapists can't really do a lot against scoliosis, but there are some smaller things that can be done on children with severe hypotonia. Sport, for example, any kind of motor activity is good as long as the child likes it, as long as the child enjoys it. Since early childhood, it is really important to do sport, regardless of the type of sport. Here in this slide, you can see many different types of sport. There is no side effect to sport to be done with the family or in associations. Then I know that sometimes it is not easy to find inclusive clubs when children are grown up. So it is not easy to find a sport activity that is tailored to the needs of your child in his stage, in his situation. And because sometimes children are not interested in competition. So I know that. Then, well, what are the qualities of a good kinesiologist? A good kinesiologist needs to be optimistic, a positive attitude, and it takes a lot of effort to do sport. We need to be aware of that. It's as if you were carrying a 10-kilo bag on your back. This is hypotonia. So your children with hypotonia have the impression that they are carrying a 10-kilo bag on their shoulders. So it's not easy for them. So it is important to be respectful. Every day can be different. And it is important to encourage children to be patient and to motivate children and also to be creative. For me, this is really important because otherwise they get bored, and we also do get bored. And being respectful means that we need to be aware of the fact that children also do many, many other things. They don't just go to the kinesiologist. In conclusion, what is important to me is to understand really what are the current motor needs of our children, because parents are those who know their children the best in the best possible way. So what you do at home, I mean, we can't see it during the consultation. We don't really see what you do at home. So please share with us the needs of your children. And then it is important also to work on inclusion. For example, if the child wants to join a club, a sport club, well, we can work on that, because sometimes children do want to join a sport dialogue, a sport club. It is important to have an open dialogue. And I think that it is really important to exploit, to bear in mind that children really want to learn. They're interested in learning. And this is what I wanted to tell you. I could add thousands of things, but I'm looking forward to hearing your questions. Thank you very much.
Bonjour à tous. Je remercie
Hello, ladies and gentlemen. I'd like to thank the organizers to giving me the possibility to talk about learning disabilities, which is neurodevelopmental disorders,
ou qui soient neurogénétiques.
can be classical or neurogenetic. Many elements have been given by my colleague here on how to take care of the neurodevelopment that will be the base. Slight problem with the slides. The neurodevelopmental disorders are now really well categorized, listed from the DSM-5, so the general classification of neurodevelopmental disorders. And this allows not only to categorize, but also to find them, to identify them, and have a more precise intervention, and to be able to have a follow-up over time. Once we have understood that all these disorders can be in the same brain and on the same person, there will be a care which will be tailored ad hoc and a lot more precise than what used to do before, because we know now that, for instance, the eye-hand coordination depends also on the tonus of your muscles and the development. We will start through axial tonus and proprioception, so all these sensors, because children to be able to develop and improve are based on sensory and motor skills. That's why in this circle of categorization of these disorders, we will find children with with more intellectual difficulties, but also with children with heterogeneous difficulties, and children with cognitive difficulties as well. Here we need to understand that the categorization of these different disorders in their learning starting from 4 or 5 years of age will depend on the experiences and skills and competences, transmodal competences, from moving from a visual comprehension to a proprioceptive comprehension to a motor knowledge and comprehension. My idea was to show you how the brain works where there are no subjacent difficulties or disorders, and where the deviation happens with the insurgence, with the occurrence of disorders. With the same cerebellum plasticity, based on our environment, we will optimize the development, or on the other hand, limit the children in his own internal experience. And this allowed how we can adapt once that we have understood what the children is able to do.
Pour comprendre que le mouvement dont on vous a parlé va nous permettre.
We can understand that the movement that we talked about will allow us to have some retroaction loops.
Correcte, je vais la garder parce que dans mon cerveau j'ai une récompense.
If what I have done was correct, I will continue because my brain will produce dopamine. Or if it doesn't work, on the contrary, I will change the process to learn to do it differently. And so rapidly, in our learning, with the neurogenetic pathology, will go through the body with a motor memory and an emotional memory, and progressively verbal memory as well, with the capacity in time to have abstraction in the neurodevelopmental difficulties for the disorders we are talking about today. The capacity of abstraction can be limited either through language or through the sensory and motor representation. And it is important to avoid some comorbidity in this case, some counterproductive sensory feedback to certain movements or activities. Normally, each lobe has a function. This is just an overview, of course, is a lot more complex than that, but each lobe is important because in each multimodal and sensorial area, this will allow for complex activity. And if it's a baby with hypotonia, there are, for instance, that some tells that tell us that his learning trajectory is different, and we'll have to work on that. And the multimodal and sensory association, for instance, when the baby takes something in his hand, he can look at what he got in his hands and recreate an image of what he's looking at. And these haven't been worked a lot about that. We see in some children and some are able to do that and some other aren't, and these competences are stimulated by psychometricians that know the troubles of neurodevelopment. In this different localization, there is an important circuit, a part of our brain which is more internal, and that is in memory circuit linked in between the structure that we call amygdala and hippocampus, allowing us to specialize all execution tasks that we perform. And so the right and left brain specialize in time in different skills, and normally through development, the right hemisphere will get to maturity before the left-hand one, and will be able to associate rapidly a melody, for instance, to a movement, or for instance, the little songs that you can play and dance at the same time. So the certain music and movements are linked, and also the representation of the movement of the sequence 1, 2, 3, for instance. This is the beginning of learning on 10 and numerals. This is fundamental because for the moment, for standard learning will go through words, and for certain children, this difficulty of abstraction is a problem because they cannot visualize the counting. If it is used where there's a sensory motor support like adding or subtracting, there is immediately a visual and motor representation, allowing for the children to adapt rapidly in the space and in society, because these comprehension and learning steps will remain, because there will be some automatic skills that will develop. This is simply to show you that finally, even though our brain is really specialized,
il est tout le temps en réseau et il va largement dépasser
It
la
is always working in network, and it will go over the area that we call the reading area, the calculus area. Finally, it is good news for us because the brain zones are really specialized, but in order to be functional, we are using several circuits at the same time. So there is a capacity for adaptation, a cerebroplasticity that allows us to do this. These are a really old image of brain imagery. And you see that in, let's call it a standard adult, we do not use only the comprehension area, but we activate another part, which is a motor part for linguistics called the Broca area. And at the same time we listen and you articulate my words in the trying to finish my phrase. And that's why, because we are permanently using all these elements that in normally, and then to put ourselves in a situation to facilitate this learning, to better visualize the overview, the complete view of things. And of course, by talking to a children, we will have all his motor and intellectual skills working at the same time, trying to reproduce the words.
De neurones miroirs qui sont la base de l'apprentissage.
These mirror neural cells are important for learning, to acquire new skills and to push them to do better and better and more things. There is the same thing. You see that I could, while I speak all of these words, I only should activate my motor area for language, but instead of that, this is lit, of course, but then you see some activity in the parietal or occipital zone as well. And progressively, we learn that even when I speak a word, I'm not just speaking a word, I'm using a lot of other skills and competencies in my brain. And at the same time, I will assess, is that said well, is that not said well, and so on and so forth. Generate words, it's really fun. With an initial language, we will get fun to produce new words. And finally, it is in the motor area and the language area we have activity, and then the sensorial and motor representation. If I say mother, for instance, we all heard the same word, but we will all link it with a different smell, a different texture, a different sensation. And this is the abstraction of the word. So playing with the word, it is exactly that, because the representation of words in the abstraction will allow to facilitate automation of the process. What does that mean? The more the children can tell if it works or if it doesn't work, the more it will learn of the process they use to get to the result.
Here you can get the idea of the complexity of neurons. When you say that the brain is specialized in some specific features, that's because, I mean, thanks to the information that we have, even though a lot needs to be understood or learned. So, for example, here you can see that there are networks that are created to ensure functioning, but there are also specific relays and connections. So the brain is really plastic. that is why you need to stimulate the brain of children, because if you stimulate very early, at a very early stage, solutions are going to be found. So this is, here you can see reading. What happens when children read, 8-year-old reading, the same level of reading. And these are the networks of neurons that are created to express the same things. So the clinician will measure the same thing, and the results are really different. So term, this is a child born at term. And on the left hand, you see a preterm child with lacking stimulations, and the route is very different. To achieve the same level, the process is really different. So perhaps in this case of the preterm child, there was a lack of stimuli. When there is a mutation or an anomaly, what happens is that the brain keeps on creating links, associations. So these connections are created, but if these links are not used, such connections are eliminated. That is why it is really important to stimulate all the time. And this is where brain plasticity comes into place. And the genetic data will play a crucial role. And once one function is stable, well, the brain will work on something else. There are three types of neuroplasticity having a role in the development. One type is linked to learning new notions and information. Neurogeneticism taught us that if, for example, we have already some knowledge, for example, we already know 2 different types of vegetables, we will learn the third one much more easily. While if we do not know any vegetable, learning just about one vegetable is going to be much harder. We need to work on categories. For example, if you think that this child doesn't know any vegetable, so it will be very easy for him to learn one vegetable, well, that is not the case. And then there are other types of neuroplasticity. And in addition to that, in the maturating process of the child, this movement that I do with my mouth, this is what the child sees. Well, this you were looking at my mouth as it was moving, and the perspective of a newborn is that by looking at my mouth moving, he can learn more, because the more we are abstract, the more we are vulnerable. And this has repercussions on the behavior. Now, these elements can be observed in all children because our sensory cortex is full of associations because of the stimuli. And then there are associations that are created. For example, if you are stimulated by the language, you can keep strong associations, while associations that are not very stimulated, that are not stimulated very often, they are going to be eliminated. The frontal cortex, starting from 10 or 12 years, becomes really important because since teenage years, these children are going to have a prefrontal moment in which prefrontal connections will be created, and they will implement new strategies. So we need to make sure that our children have everything that they need, and all the skills to adapt to each situation and go to the next steps all the time. We already know what happens in premature children at pre-cortex level, and synapses are created, and this formation of synapses is quicker and quicker. It accelerates.
Et voilà. Et la mémoire, finalement, l'enjeu de ces apprentissages avec ces contraintes.
And then we know that these constraints have a role, and it's a sort of pyramid. The children are exposed to knowledge, and it is really important for them to keep information and to be able to retrieve that information. For example, it is important to say to children, well, I know that you have this information. We need to consider all the elements of memory. There are different types of memory, episodic memory, for example. For example, we tend to say, well, this child has a good memory. For example, a child is taken to a specific place and he knows the way, and since he doesn't want to go to that place, he doesn't want to take that street. That is a kind of memory. And then there is emotional memory that works really soon in newborn, standard newborn. The hippocampus is close to the amygdala, and it's a kind of memory circuit. The memory circuit plays a role in language creation and abstraction. Then the idea is to understand the most important thing is how to encourage memory. And we need to use the only circuit that we know, that is the one that you can see here. When we do a test, because we always want to know what is the situation of children with the neuro development difficulties, it is not easy to do tests. It is not easy to assess everything, to make measurements. I know that memory is really important, and sometimes you're told that your child's memory is not really good. Well, that means that the tools that we have been used, and these tests work on lexical memory and abstraction skill. So there are gestural visual memory to say, okay, we can identify this skill, even though the another kind of elaborated language is not possible. Here you can see the representation, visual representation of memory, the different types of memory with the physical memory at the bottom, because the tonus that we have, the posture, even in newborn, is visible. And mom can anticipate very soon the activity of her child. So there is a physical memory that starts from a very early age, and then a procedural memory, for example, when the child pretends to be something to do, for example, to clean, he pretends to be cleaning just as his mom is doing. And these elements are really important in this early stage. That is why, for example, people say that once you have learned to ride a bike, you will never forget about that, because these kind of skills work in a sort of circle, a circuit. But you can also have a short emotional circuit that can block you. That is why it is really important to intervene in case of problems. And then there is episodic memory that is linked to emotions. Amygdala. Amygdala is really active, and there's a lot to be learned. For example, we can say that there is amnesia, children amnesia. But the problem is that if the child doesn't have really high linguistic skills, the child will not be able to tell what is happening. But this episodic memory, for many children having language difficulties and learning difficulties, well, this episodic memory is going to be really, really useful. There is one example that I mention very often. That is the history of Nicolas, he's asked, what is the name of the river flowing in Paris? And Nicolas is at the bottom of the classroom, and he, in his head, in his mind, he is thinking of Paris with billboards and the name Seine, the name of the river. So he is looking for that information in the episodic memory to reply to a question that is related to semantic memory. So he has language-related disorders, and it is hard for him to have access to language directly. That is why he needs to go through episodic memory. So episodic memory can be really helpful. And that is why when you are observing a castle or a drawing, or you're looking at a stone, it is important to associate an emotion. Because if you just have information through abstract language, that is a problem for children, because they also need the support of other senses, and namely episodic memories. So in association with episodic memory, it's much easier for them to learn something. And then working memory. Working memory is important for us to speak, because we are in a culture that is very much based on speaking, on the language, and it is really close to amygdala. For example, an anxious child, for him it will be not easy for him to speak out, because his amygdala gives him the sensation, the feeling of anxiety. So our learning and our memory circulate all the time, and as a result, a child that reverbalizes what he understood, even though it's not easy for him, well, he can refer to his emotional or memory related to emotions. A child who is having problem in learning, well, he becomes anxious, and that is why it is important to give him back words that can help him change the circuit a bit. So it's a kind of circulation of the memory in order to have more space. So there is quite a close relationship between cognition, motivation, and emotions. We need to keep this in mind when we are with the children with the development disorders. It is important sometimes to leave the short circuit of emotions to go little by little to abstraction and keep on learning something. So this is important to have a good relationship. Each educator and parent should keep this in mind. And then these are the two circles, so long circuit, short circuits, sorry. So the stronger the stimuli is in a child, and more the stimuli is connected to an emotion, well, the better it will be for him to associate ideas. For example, the word mom will be associated to a certain emotion, while multiplication or maths, the word maths will have a negative association. Or perhaps if you have learned maths when you were doing something pleasant, well, this word will be associated to a positive emotion, and it will be easier for you to learn. That is why we need to adapt to help children to learn or to stabilize these information.
Juste finalement pour vous dire.
Finally, to say the oral language reference point, but it was more on the attention span that I wanted to talk a little bit about, because these are elements that we hear a lot of time. They say the language is a little bit developed. Written language is difficult because there are some attention span problem, or academical learning is difficult for the same reason. Attention is always linked with its circuit that we know better and better, that is represented here. Attention is well, being awake or asleep. This is the basis, and then we will have an attention that is visual in the posterior cortex, associative here, language, and then a frontal attention which has several development stages. When they are 4 years of age, you saw this, is the age when they start saying no, because they are developing their assessment area. And these attentional elements are developed through time, and this intentional circuit, when we will test, we will look at where it works and where it doesn't work. We will test its hearing attention. Is he listening all the words that we say? Can he repeat them? Can he or she, of course, listen to a music and at the same time understand some words that are spoken at the same time? Can he look and at the same time listen? And all of these attention elements are developed through and assessed through several neuropsychological tests, and we'll be able to say this is stronger in this children. We will use it as a way to go forward, or this is a little bit behind, is a little bit weak on this in this respect. We will do with something else. These can answer sometimes to treatment, and there you need to pay a lot of attention to be vigilant. Why? Because there can be some attention span difficulties simply because the stimuli is not right. If the children hasn't been tested, we do not know why. Are we saying too complicated things that transform in a lack of interest that can be mistaken as a lack of attention? Is this lifespan problem linked to fears that children might have? So when we talk about attention span problems, there is a need for a progressive exploration of the problem to understand how to take care of that and how to develop and devise a solution. These different types can be hearing, double task, impulsivity as well can be linked to this kind of troubles. Can they wait? We heard it in psychomotricity. Can you count till 5, till 4? We'll see if they can delay their response time. And we will also try to see how long they can maintain their attention. And if they are able to do that, we will use this to teach them further things, something else. And otherwise, we will be talking about hyperactivity or lack of attention or lack of attention span disorder. But we need to assess the situation because there can be many causes to something that can look at first as a lack of attention span. On these disorders, there can be comorbidities, and sometimes are children that are not right that present this kind of disorder is because the children can be depressed. The circuit can manifest through a attention span disorder that are difficult to assess and to diagnose for experts as well. So we will look for other tells, position, anxious behavior, learning disorders, particular situations or not. Here are some elements to say that when we decide on a treatment or for certain types of treatment, this was assessed and we looked on how it could modify the attention circuit.
Donc, finalement, on va avoir des troubles d'apprentissage.
So finally, we will have some learning disorder which are implicit. For instance, we do not learn to open a door. They say that you cannot learn how to open a door, but with a certain kid with dyspraxia, for instance, it is impossible for them to create the chain of movements that you need to open the door, to take the handle, push it, and so on and so forth. And so some motor problem are linked to are part, of what we call implicit learning disorder. We do not learn them as we would learn something academically. And this is based to a sensory disorder in the infant. And so we have to pay attention to hearing, to the coordination in between hands and eye. And starting from then, we can start to build up more and more complex movements and things. And of course, motricity. When they say, when my colleagues said before, we will do some bends, some flexing, and we will do a series of several of these. The more we do this, the more we improve the capacity to keep the representation of the quantity of the children. So these elements of calculus or imagining are developed through motricity because it doesn't need other tools. It can represent itself the movement or the situation. When we have children with these difficulties, but I think you talked about this morning, we need to be several experts to assess and evaluate the multiple difficulties, but also the resources of the children, because during the development we will, I don't like the word, but to try to optimize all this and try to avoid the creation of comorbidities or linked problems. But the better you get, the better you can learn new things. It's really difficult to start, but it becomes more and more easy as we go along. And it is important because otherwise we are always overcharging the short circuit of emotion. So we need several experts to work with the children when we have discovered this psychomotricity problem with all the support that you probably talked about before. I will stop here. Thank you very much for your attention.
Thank you for the invitation. I am going to speak about a nutritional aspect in and syndromic obesity. We are going to speak about nutritional aspects, and I am an endocrinologist, and I work at the Centre de Référence Maladies Rares, Prador, and I mainly work with adults, so starting from 18 years old. So I have more experience with adults, but if you have any questions about children, I can try to answer. Okay, what are we talking about? We're talking about obesity. There is standard obesity, most of the cases, and it's the result of the interaction between genes and the environment. So there are variants interacting between them, and that is why we speak about polygenic obesity. So it means that children inherit from their parents a certain a disposition, some specific genes. Then in some genes there are chromosomes that carry them a higher risk to become obese at an early stage. We have very many genes. Last year, an article was released, and we called it obesity, rare forms of obesity, in which we talk about all types of obesity. There's something wrong in my slides, but never mind. Okay, so there are syndromic forms of obesity and genetic forms of obesity. We talk in this article about how to diagnose such forms of obesity. Then there are different syndromes, so the SPW, X fragile, and the BB5. There are different genes that play a role in obesity. In order to diagnose rare forms of obesity, there is a clinical screening, and very often the level of severity of obesity is quite higher. And there are some genetic forms of obesity that are connected with neurodevelopmental disorders. In all these syndromes, there is also neurodevelopment disorder. And then there is also an endocrinous deficiency. So the regulation of hypothalamus, the hypothalamus plays a role in the activation of hormonal functions. So there could be also other symptoms, for example, in connection with the regulation of sleep. When we think that there is a rare genetic form of obesity, well, this is and it is an early form of obesity. So we make an assessment, body mass index, so the ratio between the length and the weight. So you can see the curve of standard children. This is the standard values of standard children. And perhaps you know that the definition for adults of obesity is for the body mass index that is higher than 30. So we draw this curve for children, and we try to understand if this form of obesity has started soon and if there are any changes in the growth as the child grows up. Here you can see a curve that starts very, very soon. It is not standard at all. This was not a patient with MYT1L, but once again, it is a really severe form of obesity because when this person is 19 years old, the body mass index is 59. And when you have a body mass index that is higher than 35, that is already very severe. And rare forms of obesity are connected with problems in the behaviors, eating problems, eating behavior problems. So in international literature, it emerges that on a cohort of people between 1.6 and 34 years and with a median age of 8.4, 59% of people are overweight or obese and 45 have eating behavior problem. These are individual curves, and this was the beginning of these assessments to start at 3.5 years old. So we are really in the criteria. We have the criteria to define a syndromatic rare obesity. So here you can see a simplification of the hypothalamus that is in the brain. There are different nucleus, but the most important one that comes into play in this form of obesity is the paraventricular nucleus. In this nucleus, there are some neurons that produce neurotransmitters, and such neurotransmitters act on receptors to give functions, mainly the regulation of energy balance. What does it mean? The regulation of food intake and energy consumption. In orange, you can see anorexigenic channel. So in this case, there is much energy, more energy consumption and less food intake. And as a result, food intake is standard. So you eat on the basis of what you need. And then there is orexigen neurons that stimulate, for example, NPY-AG, RP. These are hormones that increase food intake and decrease energy consumption. There are many weird names that you can hear in this slide, but what you need to bear in mind is leptin, that is a hormone produced by the fat tissues, and they signal to the brain when there are reserves. So here you can see the receptor, and when everything works well, there is POMC hormone here, another hormone MSH, and so when they are in connection with MC4R, the food intake and energy consumption is regulated, and so it means that you eat on the basis of what you need to eat. Then you can see many other names, SIM1 and others, MYT1L. Well, we do not have a lot of information. We have much more information about zebrafish rather than humans, but we know that leptin plays a role, especially in connection with MC4R. Okay, then disorders in eating behaviors. We know that hypothalamus regulates food intake that give us the feeling that we are no longer hungry. So you eat and then you feel full. So it means that you're no longer hungry until the next meal. But when the hypothalamus does not work well, you do not feel full, and so you are pushed to eat more, and this is hypophagia. And then there is the rewarding system that is regulated by many factors, oxytocin, dopamine, and other hormones. So that is very much connected with hypothalamus and especially alpha message that I mentioned. So this rewarding system, when it does not work well, where there is a situation of addiction, and food addiction is just like alcohol addiction. There is Prader-Willi factor, and in MRI, we can see that some areas of the brain are activated when people having this disorder see food. And it's a form of obsession, food obsession. And so the person looks for strategies to get food. So even robbing food, stealing food, and then intolerance to frustration. The interpreter apologizes, but the speaker is really speaking fast. In any case, when the person wants food and is really looking for food and the food is not there, the system is no longer working well. And then there is the cortex. The cortex can be inhibited. So the cortex help us to have self-control on many types of behaviors, including eating. And For example, when we need to make a decision to feel good, we need to make a balance between having an immediate pleasure and having a future benefit. In these kinds of disorders, there is a lack of self-control because the cortex is not inhibited, so we can't really make good decisions. And the hypothalamus is altered. And this explains behaviors. When we speak about eating behaviors, we also need to bear in mind some organic factors, but there are also other factors that come into play. That is the case for people with standard obesity. For example, emotional factors play a crucial role. For example, stress, stigmatization, and boredom, emotional dysregulation. These emotional factors play a crucial role. Any traumatic experience is going to play a crucial role. And then there is also external factors, for example, family habits, and then for example, if the fridge is closed, restrictions imposed by somebody else, cultural norms. So if you do not have access to food, the behavior improves. The behavior can improve when the environment is less stressful, when there are activities, when stress is reduced. And in neurodevelopmental disorders, there are also psychotropes, neuroleptic, especially that regulate the areas of the brain that I've mentioned that are going to activate, to have a role in the regulation that is already disrupted. Psychotropic substances have a negative impact, and also the alteration of sleep. Eating behavior varies according and in the life, during life, and according to the different steps of life, and also in connection with emotions. Okay, the request was made to speak a bit slower because the speaker is speaking really, really, really fast, and interpreters, it's hard for the interpreter to translate everything. Okay. So how can we assess eating behavior disorders? It depends on the age. If we are speaking about the child, we talk to the family. If it is a teenager, we talk to the patient. So we take a look at the person, different factors. There are questionnaires. So we observe the person when the person is eating. And then it is really important to talk to families also with when a patient is a teenager and adults, because sometimes even if you can speak about yourself, it's much better also to listen to your family. For example, we work with a team in Toulouse, and they have a questionnaire thought for children. So to understand what is the relationship of children with food. So, for example, is the child too worried about food? Does he want to have too much food when he's served, and so on and so forth? Or does he finish his dish all the time, all the time? Or does he want to finish somebody else's food? So these are examples of alterations. Does he hide to eat? Or does he create stock of food? And is the access to food controlled? because if that is the case, all these behaviors are going to be controlled. So this is a questionnaire that can be used when interviewing families. I also find a sort of questionnaire when I interview adults, in particular the Dykins questionnaire that was drafted for people with Prader-Willis Willi disorder when but this questionnaire can also be used with people with intellectual disability. This questionnaire has 3 dimensions, for example, one about behavior, impulsiveness associated to the request of food. I'm not going to read through all these questions, but there are 5 questions about the relationship between the person and food.
Que j'ai pas vraiment traduit en français.
There's a dimension that is called drive in English. It is the imperiosity, the need, the will of talking or consuming some food stuff, and the difficulty to pay attention to something else. What happens when we refuse to give the children food? Does it get angry? Is the children or does the children or the young adult perseverate to try and find food or getting second or third helpings or so on and so forth? This is the drive dimension that we see quite often in these kind of disorder. And then the severity dimension. So how is the person living the situation about food on a daily basis? Are there any particular behaviors, as any food intake in between meals? And what interferes with the normal life of the children or the adult, like school activities and so on and so forth. We will assess and evaluate all these dimensions with the Dykins questionnaire, allowing it also to assess the evolution of the disorder over time. When we have a stable way, the food intake is equal to the spending given by the base metabolism. This is linked to the muscle mass. And when we have a syndromic obesity, we have intakes that are linked, especially with the with the bin full filling. We have an intake that is superior to our spending. Either we have an hypotonia in terms of muscle, which means that the base metabolism will be diminished, will be lower, and physical stresses are also lowered, are also contained. There is, most of the time, a sedentarity and a difficulty to move. That is also in the motivation.
En fait, de se mettre en mouvement. Donc, c'est en fait, quand on va déterminer un petit peu la prise en charge.
When we will determine how to follow the patient, how to help the patient.
C'est tous ces aspects-là qu'on va prendre en charge.
Or we will take into consideration all these aspects. Just a couple of words on why do we try and treat weight problem. We're not talking about aesthetics here. These are really health problems because there are complications linked to obesity. These complications will appear during adolescence and when adults. These are rare in children, but to avoid the insurgence of obesity, We need to take care of the situation as soon as possible to avoid the situation getting more and more severe. The comorbidities that we see most of the time are metabolic with diabetes, the NAFLD, hepatitis, these are really frequent when we have weight problem, and then cardiovascular complaints. Something else that we see quite a lot are sleep apneas that can lead to hyperventilation asthma, thromboembolic like phlebites and pulmonary embolia, and functional complication like arthros, osteonecrose, and venous and dermatologic complaints as well, psychological as well. It is really important to see because there is an alteration of the hypothalamus here, and these can reinforce by psychological troubles, especially with discrimination and self-esteem. And when we have important weight problems, we normally have a low self-esteem.
Puis toutes les autres que j'ai listées, je ne les détaille pas, mais c'est pour vous montrer que quand on va évaluer ces personnes qui ont des problèmes.
When we will evaluate these problems, of course, we will be talking about weight, but the priority is to assess all of these complications and to treat them.
Donc, si je résume par rapport à la situation...
If I summarize in respect to the medical situation, syndromic obesities are complex situation because we have several different disorders linked together, some linked to the syndrome, like orthopedic malformation, neurosensory complaints are added to the weight problem. We have problem in terms of behavior. We talk about some of them, but the list was not extensive. But anyway, we have some neuropsychological disorders. We have difficulties, physical difficulties in moving, and a loss of autonomy. This is more true in aging people, of course, but is still an important point. And then social aspects, when we have a weight problem, these are also linked a lot, as I said previously, to the environment and the people in our entourage. So when we treat these people, we have to work on all of these social aspects as well, because we know that weight problem will become more serious in unfavorable environment. So the situation is complex, so you need a holistic, a comprehensive, a complete treatment in all of these aspects, taking care of dietary behaviors and guidance for the entourage and therapeutic training. This is done several times in with pediatrician. In adults, we have a harder time, of course, finding services able to coordinate the whole holistic approach. And as physicians in the hospital you work in is what we try to do to tackle the whole situation. It is a complex situation. You need to take into account of all of the dimensions of the problem. And as I was saying before, there is also an educational aspect. And we worked a lot these last 3 years on how to engage the entourage as well. As I was saying before, the entourage is really important to limit these behavior and to control them. And so we try to train and educate also the entourage. This is the mapping of how the treatment is organized and where it is organized. In France, we have Pradom reference centers, reference centers that you can see here, like Paris, Toulouse, and then the competence center. And then we also have specialized center for obesity, which are several times you see these are the same with the same people, the same specialists in the same center. So these specialized centers for obesity that they take care of this syndromic obesity disorder. For the last 10 minutes, we will talk about how to treat them and why are these rare in respect to common obesity, because there are recommendations on the way of life, physical activity, but we know that it works slightly less in syndromic obesity for several reasons that I'm not going to list here. But anyway, what is sure is that we need to find new approaches and especially adapt the treatment to the different kind of disorders and the comorbidities, and adapt all the whole process, even the physical activity. This is an extract of the PNDS, the National Diagnostic Program that I have coordinated and was published in 2021. Where we create practical dashboards where we list the targets, the objectives, and we make also a list of all the tools, because of course you will not use the same processes for an adult and for a baby. I will show you an example here is for adults. I will not talk about all of them because we do not have the time, but you can have these slides and the PNDS in the completed text. You have these dashboards for all ages. The objectives are to create a dietary framework, ritualize the meals, and no doubt and no hopes, the meal is defined as it's defined at the proper hours. You need to increase the volume of the meal and decrease the energy intake. And for that, we use some system called FALC system. And there is where you really need to engage the entourage. You need to identify possible sources of intake. You need to take care to surveil the fridge, the money the person has that can be a source of foodstuffs. And then you need to explain all the modalities, for instance, in the
des choses comme ça, parce qu'il faut que tout le monde soit très cohérent.
People of the entourage of the daily life of the person. He hasn't to have possibility to buy food or to get food. This is the base protocol. Then, of course, we need to control the behavior by lowering the anxiety and impulsivity in respect to the food stuff. In order to do that, you need to create a calm and trustworthy environment.
L'enfant ou l'adolescent, voire la personne.
And try to calm the children or the person when they start being frustrated.
Parce que c'est une escalade, hein, je vous ai parlé d'addiction. Donc l'escalade en termes
this is an escalation in terms of you need to anticipate festive moment, negotiate modification, protect from impulsivity, for instance, of food intake between meals and so on and so forth. Ritualization is really important and allow to protect oneself from impulsivity. And then we have some cooking courses, especially with the Prader-Willi syndrome, where there is an addiction with the visualization of food, but maybe on other syndromic obesities, these cooking courses are not so important. And then we need to favor the coordination and coherence in between the different stakeholders with the different institutions. For the physical activity, there are some recommendations, which are the following. Adults should do in between 150 and 300 minutes in terms of sport activity with a moderate intensity, let's say like swift march. And you need also to add some muscular strengthening activities. These are recommendations that are given in common obesity, not only in these kind of cases. We know that dietary intervention and physical activity have an incidence in terms of weight loss. We're talking about 5%, and 5% is already good because it eliminates some of the comorbidities, but sometimes you need specific tools. Documents have to be in fog. You need to train the personnel. You need the support of the entourage, behavioral strategies, and adapted processes. And when we do not have all these tools, all this goes against the physical activity. So in the PNDS, we put some adaptation that are recommended in case of this obesity with a neurodevelopmental disorder. I'm not going to list them because we do not have time for that, but if you are interested, go to and see. You have to foster amusing activities that the person will want to continue. And also you need individual sessions. Of course, hopefully these are not taken charge by the national health system, at least here in France. We are far from the individual care that we would need. I carried out a research protocol for Prader-Willi, and in 16 weeks of physical activities at the domicile of the person and working with them individually, we have seen outstanding results in the lowering on hyperphagia. We could stabilize the weight and improve the quality of life. We also measure the physical activity with an activity meter, and we've seen an increase of physical activities in between sessions. That means that when after a sort of coaching, this creates a virtual circle where the person will start at one point to be willing to have a physical activity on their own, even though the coach is not there. So this is FOX, ritual planning, some visual tools. Once again, you need to engage the entourage and foster the progress. This is, for instance, our the advices we give in some of these processes. You'll be able to read them later on. So physical activities on a daily basis, the role of parents, for instance, taking the dog out, or so on and so forth. Institutions. It has to be for everybody, not only for the young obese person. And then the role of parents and institution, and break the sedentarity with the role of everybody in the entourage to start this.
Les traitements.
Okay, let me finish with the treatment, because for 3 or 4 years ago, we started having the opportunity to have targeted treatments. So this is the schema of leptin melatonin. So this MC4R is a receptor, and we have now antagonists of MC4R that we can use. Imcivree, that is the name of the treatment, can be used here. This is a study with 10 patients and the second one with 11. And you can see that we speak, this is the median age, 18.4 years, and the mean age in the second case is 23.7. And you can see the percentage of weight loss, so minus 25.6. And 80% reached had a weight loss of at least 10%. And in the second case with the LEPR, there was a weight loss of minus 12.5% in 1 year. So even though this weight loss was slightly smaller, still there was an improvement in the life living standard. This is the difference in 1 year in adults and in children. So when you improve the quality of life, this improvement can be observed in patients, but also in the family. And these are the results for 7 years, long term. So with patients with melanotin, this is the weight change that decreased dramatically, and then it remained stable. One person lost 55.6 kilos. This was a really severe obesity. I do not have results for children, but I'm speaking about really severe forms of obesity in this case. And there was a dramatic improving life in quality of life. And in Bardet-Biedl syndrome, that is a syndromic form of obesity, there is an alteration at the level of hypothalamus with the babysome complex that is mutated. There is a mutation that interacts with the LEPR and MC4R. So they have an interaction, and this situation results in weight gain before 5 years old, and 80% of adults having Bardet-Biedl, even 84% are overweight or obese. These patients have been treated by Imcivree, and you can see the results after 1 year of treatment, the mean age 20 years, and the body mass index for about 42, so grade 3, really severe form of obesity. And these are the results for patients more than 18 years old and for teenagers. So you can see the weight loss minus 9.5 in 1 year. There are many variables, many differences between individuals or among individuals. Second bottom part of the graph results in teenagers. In general, what we see here is actually a worsening of body mass index. This is the tolerance of melatonin in patients with BBS. Alpha message, that is the hormone that plays a role, pigments the skin with receptors that are on the skin, and sometimes there is an irritation on the screen, on the skin, and there could also be side effects related to digestions. And these side effects decrease over time. All these result in clinical trials led France and institutions to authorize the put in the market. This treatment is now available even before finishing all the procedure of the clinical study for adults, but also children having Bardet-Biedl syndrome. In this specific syndrome, there is not no authorization and no study showing the effectiveness of this therapy. Another study is going to start, a clinical trial is going to start soon with the variables. It does not include MYT1L. So we need to wait if there are other applications, other applications of this therapy. And then there are also other therapies that are available in cases of obesity. Semaglutide, that is an antagonist, an hormone antagonist to hormone GLP-1, and as a result there will be a reduced appetite and cravings, and also improved control of eating. It reduces energy intake and body weight. In standard obesity, now I'm not speaking about syndromic obesity, in standard obesity, you can see that semaglutide enables about 15% weight loss. When you take a look at these weight loss in patients losing weight, you can see here the results between semaglutide patients and patients with placebo. So this is a really interesting therapy that can be really effective with the standard obesity, and as a result, the French authority is going to authorize Wegovy, so semaglutide, for people with body mass index of more than 40%, with hypertension and the cardiovascular disease, just for adults for the moment. In syndromic obesity, the assessment hasn't been made yet, so we don't know if the results will be the same. When we have Bardet-Biedl, we use a therapy. Then I've talked about the second study. We don't know what will come next, and we'll try to have as much information as possible for to have a therapy for syndromic obesity. Then side effects. They are they are really mild. They can be annoying, but they are mild. For example, problems, gastrointestinal disorders, diarrhea, nausea, vomiting, and but these side effects disappear over time, and then there is an escalation of doses. So from 0.25 to 2.4, this is the dosage. And as for cardiovascular disease, the risk is really mild, because when you start giving a drug that has an impact on behavior, well, you really need to be careful, because in some cases there were problems at psychiatric of psychiatric type. My presentation is over. Now I am available to answer your questions if you have